Good day again. This is Jim Pickett from IRMA, and it looks like we're hitting critical mass, so we are going to get started with our webinar today. Again, this is Expanding Choices. I'll look into the Population Council's MPT product pipeline. Thank you so much for joining us today. A little bit of background, a little bit of housekeeping, and then I'm going to do a quick introduction, and then we'll turn it over to our wonderful colleagues and friends at Pop Council to talk us through their very exciting pipeline. First of all, everyone is on global mute. This is so we can have a nice clean recording. The slides and the recording from this webinar will be available on the IRMA website in the coming days, and we'll send everyone an email with the link to that. So don't worry about not being able to have access to the slides. We do really want to have conversation and questions, so feel free to leave questions or comments in the chat box. If you're on the webinar platform, in the left-hand corner, there's a chat box. You can leave questions and comments. We'll also be doing questions and comments verbally, and we'll let you know how to unmute your line when it comes time for that. Or if you would like, you can email questions and comments to IRMA's website, IRMA's email address, which is rectalmicro, all one word, rectalmicro@gmail.com. I would be remiss if I didn't thank upfront AVAC for all their support in pulling together this webinar. We partner with them on our webinars and are very grateful for that. And a special thanks to Levi Butcher, who is handling all the behind-the-scenes controls and making this as seamless and flawless as possible. So thank you so much, Levi. Now for some just sort of brief remarks I would like to make about the importance of this webinar and what it represents. As everyone knows, the HIV prevention trial networks, the various networks that conduct this research are being recompeted. There was a funding opportunity announcement released in late January that lays out the framework for this competition. Folks who are competing are now getting ready and starting to write their applications, which are due in early August. Actually, the date is the 1st of August when they are due. So work is already underway. And this is really critical. This is going to lay out the bulk of what's going to happen in HIV prevention research on the globe from 2021 to 2027. So things like MPTs are incredibly important. And as we've heard over the last couple of years, the NIH has not shown a lot of interest in things that are not long-acting and systemic. They've really put a lot of focus there, and we've been pushing back. Many advocates and researchers have been pushing back to say long-acting and systemic are great to have. We would love to have options like that, but we also need things that are short-acting, things that are localized, that are not systemic, things that are user-controlled and not necessarily delivered in a clinic like an injection would be or an implant would be. We need a range of things, and we've been pushing that for some time. So we were very grateful to hear or to see when the funding opportunity announcement came out that there is language in there that does leave room for the things that we care about, for things that are not just long-acting and systemic. Of course, we care about those, but we, again, want the full mix. I want to just read off a couple of key sections of the FOA, the funding opportunity announcement, just to put them in your head as we move forward with this webinar to really contextualize why it is so important that we have a range of methods, a range of things that provide protection against HIV, and in the case of MPTs, against other things, unwanted pregnancy and STIs, that are all really important to our communities. So some key language in the FOA. One sentence is, the research must focus on advancing a range of products and strategies to meet the needs of populations vulnerable to HIV infection. So that's directly from their language. I would say that if we're going to meet the needs of various populations vulnerable, we need to give them choices. Later in the FOA, they say products that do not achieve complete systemic protection but do address an agreed upon compelling specific public health need can be considered on a case-by-case basis if the scientific data supporting moving them forward to clinical testing. So if there is enough scientific data in their view. So I think we have, that sounds good, but we would want to have more discussion about what how you determine what a compelling specific public health need is, and what is the data that will be looked at, and how will someone determine whether that is appropriate enough or not. Now, I think a key section, especially related to this webinar today, is a section they talk about multipurpose prevention technologies. And this is a quote from them again, from the NIH in the FOA. MPTs may be an important strategy to prevent HIV, especially in adolescents and young women. The ability to offer effective contraception in combination with an HIV prevention product may increase the product desirability and uptake by meeting both critical needs simultaneously. MPTs might also include prevention of other STIs. The choice of which STI to target should be based upon prevalence in the target population and potential public health impact. So really, really critical. They talk a bit about the importance of integrated biomedical and socio-behavioral prevention strategies. And they really also have great language around needing to have early stage acceptability, desirability research to optimize and select candidate products and delivery systems, and to have a critical need, and that there's a critical need, to determine end user preference characteristics. And so using methodologies to determine that. All of these things really leave groundwork for us to make sure that choice is involved in the pipeline and that we have these sorts of things that we've been talking about. It's not going to happen automatically, though. I think we all really need to be engaged in this conversation and talking to leadership of the HPTN, namely Drs. Wafa El-Sadr and Dr. Myron Cohen, who are likely to be leading the network in the new era and starting in 2021. They need to be hearing from us that these are important and that we want these to be included in their application, and we expect them to be included. We expect to see research continue. And I think there's a couple of, and I'll close my remarks by saying there's a couple of opportunities coming up very soon to have these questions with a lot of folks in person. Many folks will be attending CROI 2019, which starts this weekend and early next week in Seattle. If you are going, please look for me. I'd love to say hi. And let's have conversations about this with all kinds of folks to make sure that people are hearing from communities and from advocates and from scientists what our expectations are and what we hope to see in this application and moving forward in the actual research when it begins and continues, I should say. And also for many folks in the U.S. maybe attending the CDC Prevention Conference, which is later in March. This is not typically attended by lots and lots of prevention researchers, but there will be a lot of key folks there. And I think important to have these conversations and build more momentum around this discussion and to make sure that we have what we want to see in this application, that it will be due to the NIH on August 1st. So basically the timeline will be applications are due in August. For about 8-9 months, the NIH will be reviewing them. Folks will find out early next year what the determination was, and then they'll be starting work for 2021. Now, you've heard enough from me. It's been a lot of talking from me, and we want to hear from our colleagues at Population Council. So I would like to start by introducing Barbara Friedland from Pop Council. She's going to be kicking off this webinar. She'll tell you more about herself. And Barbara, please make sure to *7 to unmute your phone and remind me when to advance your slides.
Great. Thank you so much, Jim, for that introduction. Good morning, good afternoon, good evening to everybody who's on the line. Thank you so much for joining us today. We're going to spend the next half hour or so talking about the Population Council's research and development strategy and our MPT product pipeline. So I guess a word about myself. I've been a researcher at the Population Council since the late 90s, working on microbicides development for that whole time, both in the context of clinical studies as well as behavioral research. Before we get started, I wanted to just define MPT the way the Council has defined it, which sounds promising that it overlaps somewhat with what is in the NIH FOA. So basically, we consider an MPT to be any product designed to target more than one indication, including HIV or other STDs or contraception, so any combination of HIV, STIs, and contraception. Next slide, please. So for those of you who may not know us, the Population Council is an international nonprofit research institution. We conduct social and behavioral science research as well as biomedical research. Our research is used to improve policies, programs, and technologies around the world. Next slide. For more than 65 years, the Council has given voice and visibility to the world's most vulnerable people. Our research increases awareness of the problems they face and offers evidence-based solutions, including better policies, programs, and technologies. Our research is focused in three main areas: reproductive health, HIV and AIDS, and poverty, gender, and youth. Next slide, please. So a lot of what we do is developing biomedical innovations. So we have a Center for Biomedical Research, also known as CBR, which is located at the Rockefeller University campus in New York City. For more than 60 years, research conducted at CBR has addressed critical questions in reproductive health and supported the development of innovative products that help protect the health and well-being of millions of people worldwide. The Council has an unparalleled track record of success for bringing new contraceptive technologies to market, from basic research on reproductive biology through FDA approval and introduction of new products. Our current estimate is that more than 170 million worldwide are using highly effective contraceptives developed by the Council or based on our technologies, including the copper IUD Paragard, contraceptive implants Norplant and Jadel, the intrauterine system Mirena, and the two intravaginal rings, Produring, and soon to be on the market, Annovera one-year contraceptive ring. Our research shows that women want and need a variety of contraceptive options throughout their reproductive years, and we know that expanding choices enables more women to better meet their needs and have better health outcomes. Next slide. So building on our contraceptive development history, we started working on development of microbicides at the end of the late 80s and the early 1990s. So the Population Council was one of the pioneering groups advocating for female-initiated HIV prevention options in the late 80s and early 90s. Council scientists collaborated with a group of international women's health advocates to advocate for the development of microbicides, products that women could use discreetly that would not interfere with sex, that would not require negotiation with their partners, and would not impact their fertility. We conducted a number of studies of hypothetical acceptability of such products among women and male partners. One very important study that we did early on in the mid-1990s that was published in 1998, we refer to as our formulation preferences study. We enrolled women in 5 different countries who used 3 different over-the-counter formulations of nonoxynol 9, a gel, a suppository, and a film, to see if there was a preference for one of the formulations over the others. And we found that there was no single winner, but that the suppository was less desirable than the gel or the film. These results directly informed our development of a gel formulation of Paragard, which was our lead candidate product at the time. Furthermore, we listened to the women in the phase 1 trials of Paragard who said the volume of gel was too much. So this led us to reduce the dose from 5 mL to 4 mL from the phase 1 trials and going into phase 2 and 3 trials. So although Paragard was not found to be effective in preventing HIV, it did provide us with much understanding about women, the complexities of designing and developing interventions that they need and want, and the importance of providing choice and control. Next slide, please. Today, we are continuing to understand preferences and the needs of women, people who might use our HIV prevention and family planning technologies. And this understanding is influencing, it continues to influence our product portfolio. For those of you who may have attended the HIV R4P meeting, you might have seen a preview of some of our recent findings around our efforts to understand women's preferences around multipurpose prevention technologies. In 2017, we launched a global online survey called Share, Learn, Shape, and the homepage is down on the left over there, which is helping us to understand if women would be interested in using HIV/STI prevention product that also, if it also prevents pregnancy. So I want to give a shout out now to Jim Pickett and Kate Guthrie, who both collaborated with us on developing and distributing the survey. So Jim was very involved and helpful in helping us design and get this off the ground. So the main questions that we were asking in the survey is, would women be more interested in using an HIV or STI prevention method that was also a contraceptive? Which specific products would women be interested in using? Which product characteristics are most important for method choice? And are there any characteristics of products that would lead women not to use the product? So the survey, which we closed at the end of last year, there were over 700 women who responded from more than 50 countries. And of these 700 women, 80% completed the survey. And as you can see here, the one country with the most responses was South Africa, which had 39% of the respondents. Next slide, please. So the survey found that most women want choices. As a contraceptive developer, this finding was not surprising, and we've seen similar evidence in our other bodies of social and behavioral research. We know that women want and need different options at different points in their lives. If you look at this graph here, you'll see that there's a range of interest in the 9 different formulations we asked about, ranging from 64% on the left to 48% on the right. So pretty much at least half of the women were interested in any of these products. And in fact, 94% of the respondents were interested in at least one of the products. We asked the question about whether there would be more preference for a product that also provided contraceptive properties, and 83% of the women said that they would prefer an HIV/STI prevention method that also protects against unintended pregnancy. And I think this is extremely good evidence for the language in the FOA. Again, not surprising, the number one reason women gave for not wanting to use a particular product was side effects. Additional reasons for not wanting to use something were cost, which 36% of the women mentioned, that it requires a prescription, and that it reduces sexual pleasure. So again, from what we know from the contraceptive experiences of women, this is not so surprising. Next slide, please. So we wanted to see if we could look at what predicted interest in an on-demand method, such as the gel, film, FDI, fast dissolve insert, or the gel before sex or after sex, versus long-acting methods like implants or injectables. And the biggest predictor was previous use of contraception. Again, not so surprising. So if you look on the left side, you see the on-demand methods that women who previously used female condoms or withdrawal to prevent pregnancy or were interested in a CMPT were more likely to be interested in on-demand methods. Interestingly, characteristics associated with not being interested in an on-demand method were having only one partner to prevent HIV or STIs, having used male condoms for contraception, having used vaginal rings for contraception, or not using any method for HIV/STI prevention. And then in terms of the long-acting methods, again, not surprising at all, women who had used implants or injectables were much more interested in long-acting methods than women who had not used those methods. And then there were also some interesting demographic characteristics of the women interested in long-acting methods. Those who didn't finish high school versus finished high school were twice as likely to want a long-acting method, and women 18 to 24 were more interested in a long-acting method than those over 25. We're continuing to do analysis now where we're trying to use latent class analysis to see if we can identify clusters of women with similar characteristics to help to predict interest in these specific products or product classes. So stay tuned for more on that once we have a chance to complete working on the data. And now I'd like to turn this over to my colleague Tom Zydowsky, who is going to talk specifically about our product pipeline.
Okay, well, thank you, Barbara, and thanks to everyone who's on the webinar today. My name is Tom Zydowsky. I've been to council a little less than 12 years. Trained as an organic chemist, PhD organic chemist, and also regulatory affairs writing. Since I've been at the before joining the council, I was primarily doing drug discovery work, and since I joined the council, it's been primarily drug development. So I've looked at both ends of the kind of drug development cycle. Barbara just told you about all the work that she and other colleagues at the council and across the world have done to understand what the users want and need in a microbicide product. And now I'm going to tell you how that work and the results of their studies informs our product development strategy. So Barbara showed you one example of what the council is working to do, which is to keep the wants and needs of women and men at the center of our development efforts, so user-centered design and development. And we continue to find that this means providing end users, the men and women, with choice and control over their sexual and reproductive health. So in the reproductive health program, and that's one of the two programs here at CBR, we developed the field of long-acting reversible contraceptives, or LARCs, and today are also looking at other forms of LARCs and permanent contraception. As Barbara mentioned, the council developed the copper TIUD, the Jadel implant, Mirena IUS, and most recently the one-year Anavera vaginal ring system, which was the first and only FDA-approved women-controlled contraceptive vaginal system that works for 1 year. We're also developing male contraceptive products to give men the choice to play a role in family planning. We currently have a male contraceptive gel being tested in a phase 2B clinical trial. In the HIV and AIDS program, our strategy is to develop safe and effective active pharmaceutical ingredients, or APIs as we call them, and then formulate and deliver those APIs or drugs via different user-friendly delivery systems. For HIV prevention, we want to develop ARV or antiretroviral and non-ARV or non-antiretroviral based products. And I'll give you an example of those products in the following slides. So on the next slide, we've got our MPT pipeline. And we've got three classes of MPTs that we're currently interested in the program. So we've got our ARV-based PC1005 series. We've got our non-ARV-based PC6500 series. And we've got an oral contraceptive MPT concept that we've recently started development on. So, and again, I'll talk about each of these programs in the subsequent slides. So next slide, we're brief description of our PC1005, which is our ARV-based program. PC1005 was designed to be used both vaginally and rectally. In current form, it's a gel, so it's a carrageenan-based gel. It was designed to be used both vaginally and rectally, and preclinical testing shows it has activity against HIV, HSV2, and HPV in cell-based assays in animal models. So again, it has that broad spectrum activity that we want in our MPTs. PC1005 has completed a phase 1 vaginal trial and is currently in a phase 1 rectal trial, which is known as MTN-037. We expect the results from the vaginal trial were published in 2015, I think, and we expect the results from the MTN-037 trial this fall, so stay tuned for that. So far, PC1005 has been found to be well tolerated in the vaginal and the rectal trial so far. And while gels were the only delivery system tested, we're looking at other delivery systems like vaginal rings and rectal or vaginal douches to complement the gel program. So on the next slide, we've got now our PC6500, our non-ARV program. And by now, I think you've probably seen some of the recurring themes that are kind of have been maintained in our product development strategy. We're big on focusing on user-controlled products, meeting user needs. So what do men and women want, and what do they need? And finally, products that have broad spectrum activity, whether it be across multiple STIs or STIs plus contraception. So the PC6500 series is a non-ARV-based, non-antiretroviral-based program, and it contains the anti-HIV, anti-STI protein known as Griffithsin. Griffithsin was originally isolated from algae off the coast of New Zealand, but thankfully for those of us interested in microbicide development, it's now overproduced in rice and tobacco plants. So we have a ready supply of Griffithsin to put into the different products. One of the nice things about Griffithsin is that it could have reduced toxicity compared to other drugs in development or other products, and no cross-resistance with currently used HIV drugs. So that's a big plus for the Griffithsin program. PC6500, as I mentioned, has broad-spectrum activity against HIV, HSV2, and HPV in the cell-based and animal models that we typically run here at CBR. It's been tested for vaginal use in a phase 1 clinical trial and has been shown to be well tolerated with no safety concerns. For PC6500, we're exploring other delivery systems, including vaginal rings and FDIs or fast-dissolving inserts. And on the next slide, I'm going to tell you a little bit about our PC6500 Griffithsin carrageenan FDI, now again as a PrEP device for HIV and STI prevention. So let me start off by telling you a little bit about FDIs or fast-dissolving inserts. They're solid drug delivery vehicles produced by freeze-drying aqueous mixtures of drugs. So in the case of 6500, we would take an aqueous solution of Griffithsin, carrageenan, and some excipients, freeze-dry them, remove the water, and we get a nice solid dosage form. FDIs, however, they're solid outside of the vagina, outside of the rectum, but once they're inserted, they quickly dissolve to form a viscous gel, which then delivers their drugs throughout the vaginal lumen and tissue. So in the case of 6500, the insert would be inserted, it would dissolve in the vaginal fluid that are present in a woman's vagina, form a thick viscous gel that would now spread throughout the vagina and be there ready to intercept HIV or other viral pathogens when it's deposited during sex. For the users now, FDIs are inexpensive, discrete, portable, on-demand products that have been tested well in acceptability surveys, including the one Barbara mentioned, and are currently marketed for other indications. So we recently tested a PC6500 FDI in animal models of HIV, HSV2, and HPV infection. And on the next slide, I'll summarize that exciting data, which I presented at HIV R4P last October. I want to mention that this was a collaboration with our friends and collaborators at PATH in Seattle, Washington. As I mentioned in the FDI, in the previous slide, FDIs have attributes that users find appealing. From a manufacturing and preclinical testing perspective, FDIs are also very appealing. We can quickly manufacture different size FDIs, and I draw your attention to panels A, B, and C on this slide for testing in humans, macaques, and mice respectively. So again, using the same formulation, a different mold, we can make either a human, a macaque, or a mouse-size FDI. So we tested the FDIs for their ability to prevent SHIV, which is the simian human immunodeficiency virus. It's a macaque model or monkey model for HIV. HSV2, which is herpes simplex virus 2, and HPV, human papillomavirus, in animal models. So let's focus on panel B for the time being. We're now going to look at the results for anti-SHIV activity in macaques. Macaques are a specific kind of monkey, so we can think interchangeably macaques or monkeys. So in panel B, the results were 10 macaques had a Griffithsin carrageenan FDI inserted vaginally, and 4 hours later, those same macaques were challenged with a high dose of SHIV, or simian human immunodeficiency virus. The placebo control group had a carrageenan-only FDI inserted, followed by SHIV challenge 4 hours later. So now if we look at the two graphs within panel D, the y-axis is the number of HIV copies in the macaque plasma, and that indicates active SHIV infection. The higher the number of plasma RNA copies, the higher the HIV infection. And the x-axis denotes the time after SHIV challenge. And then finally, each symbol denotes a different animal. So you can see for the Griffithsin carrageenan panel, 2 out of the 10 animals have active infection. You can see basically the majority of the symbols are located there at the baseline, which indicates no infection. So we've got 8 out of 10 uninfected, 2 infected. Now if we move over to the carrageenan-only panel, we now see that all 10 of the animals were infected. All of the symbols have now, after at least 2 weeks, or by 2 weeks, have all reached, have all shown active HIV infection. And this is a very exciting result for us. Again, 8 out of 10 animals protected with the Griffiths carrageenan FDI, where 0 out of 10 animals were infected with the carrageenan-only FDI. So if we now move to panel E, which is, again, looking at the protective efficacy of the Griffiths and carrageenan FDI in a HSV2 mouse model. Again, panel E, we've got percent uninfected in the y-axis and days after virus challenge in the x-axis. If we look at the data now for in the orange line, which is the HEC FDI, so it's a placebo FDI, we see that after 8 days, 0% of the animals were infected, or in this case, 15 of 15 animals were infected. Whereas we look now at the blue line, which is the Griffiths and carrageenan FDI, after 20 days of observation, 60% of the animals were infected, which corresponds to 9 out of 15 uninfected animals, which again is a great result, and again warrants lets us know just how powerful the Griffiths and carrageenan FDI development model could be. And finally, we'll look at now panel F, which is the HPV mouse model. And here we've got on the y-axis log radians. So basically, these animals were treated with the FDI and then challenged with HPV a couple hours later. The y-axis, and then the animals after 24 hours were whole body imaged. And the results on the y-axis is now the results of that imaging. It's the log radians, and I think the most important thing to remember here is the higher the log radians, the more infection there is in that particular animal. So on the x-axis, we're looking at 3 different groups now. So the group in orange is the HEC group, and again, animals were treated with a HEC placebo, and there's a high level of infectivity or infection in those particular animals. If we look now at the animals in the blue symbols, which is the Griffiths and carrageenan FDI group, there's a significant difference between those animals and the animals that received the HEC-only FDI. And in fact, if we now look at the next set of symbols, which is the DPBS group. So in this case, those animals were treated with the FDI but received no virus challenge. They just received a DPBS solution. And you can see now that the Griffiths and carrageenan FDI, the amount of infection or the protection afforded by that FDI is about the same as though the animals didn't even receive any HPV virus challenge. So again, an exciting result. And overall, we're excited by these great data, and now are looking to get the Griffiths FDI into a phase 1 clinical trial. And I encourage anyone who's interested in collaborating with us on that trial to contact me or Barbara about how we can team up to get that Griffiths and carrageenan FDI into a phase 1 trial. So on the next slide, I want to talk about our final development program, which is contraceptive MPTs. We've got 2 series of contraceptive MPTs currently. The first now is a combination of Truvada and a combined oral contraceptive, which we're working on with CIF funding, which is Children's Infant Fund Foundation. They're a UK-based organization. And this MPT combines 2 approved and well-established products, Truvada, which is for HIV PrEP, and the combined oral contraception, which is for pregnancy prevention. This product has a compressed development timeline and compared to a product that contains new products. We also think it will be a straightforward regulatory submission. Most importantly, we are not anticipating a full phase 3 clinical trial for HIV prevention. The second series is an exciting product using Griffithsin, which I talked about before, as part of our PC6500 series plus a non-hormonal female contraceptive. This would be another option for women, and it's an early phase development, so I don't have much more to tell you beyond that it will be a Griffithson plus a non-hormonal on-demand female contraceptive. So on the next slide, we hope that you've gained, Barbara and I hope that you've gained an understanding about the Population Council, about our pipeline, and about the research that is informing that pipeline. Barbara and I talked about user needs, user control, and choice in products. We know that when it comes to their sexual and reproductive health, women and men do want options. Options across their entire reproductive and sexually active lifetime. So our goal at the Population Council is to make sure that our products are configured to meet the evolving needs of men and women. I encourage you to contact Barbara with any questions or comments beyond what we'll discuss at the end of the webinar. And now I will hand it over to Jim Pickett for the next steps.
Thank you so much, Tom and Barbara, for a really wonderful set of presentations, very interesting and thought-provoking. We have lots of time now for questions and answers and comments. We do have something that has come in through the chat, so I will ask that question first. I have a bunch of questions, but I also want to hopefully hear from a lot of other folks. So I'm going to tell you how to unmute your phone as you're devising your question or comment. When you are ready to do that, you'll press *7 to unmute your phone. We'll get to you. You'll ask your question. And then when you're done speaking, we'll ask you to *6 to mute your phone again so we can maintain a really clean recording. So I think this is for you, Tom, or it could be either. You can both speak to this. But this comes in from Anna Forbes, and she's asking on the chat about rectal products that have been tested for acceptability by participants. Have they been used rectally by both women and men, or just by men?
Okay, I think I'll let Barbara answer that because she's the clinical expert for the MTN-037 trial.
Thanks for that question. So in this particular study, it's a dose-ranging study, so it's the very, very first study, and it's enrolling all men, women, cisgender, transgender. It's not a lot of participants, but there was no limitation on gender in terms of who was enrolled in the study. But it's the acceptability is more tolerability in this first study because they're just getting one dose of three different amounts at 3 different points, and they're not using it during sex or using it regularly at this point. So all genders and no not detailed acceptability yet.
Thank you, Barbara. We have a couple more questions coming in from the chat. I'll take one from the chat, and then we'll hold to see if someone wants to speak up verbally on the line. So this is a question that I also had. Could you tell us what the presentation of the combined Truvada will be? 1 combined pill, 2 pills in 1 casing? What are your thoughts on how that's going to look?
Thanks for that question. Yes, so ultimately our goal would be to have 1 pill co-formulated with Truvada and the COC. And the idea is that it would, again, ideally come in some kind of packaging that looks more like birth control contraception, so that hopefully that would overcome some of the stigma of using Truvada for PrEP. So that's, down the line. What we're currently planning is a small pilot acceptability study in which we would co-package into 1 capsule Truvada and a COC. So it'll still be in 1 pill. It would be bigger than the ultimate pill, but because it's a gelatin capsule and it's commonly used size for antibiotics or other things, we're hoping that it will still give us enough information about whether people would prefer to take 1 pill that has both of these products in it versus 2 separate pills.
Thank you, Barbara. As I mentioned, we have more questions in the chat, but we're going to pause to see if anyone wants to ask a question verbally. If so, press *7 to unmute your phone and identify yourself. And we'll pause. No one wants to share their beautiful voice with us today? Okay, not yet. We do have more questions. This comes from Ken Mayer. He says this, given the inactivity of carrageenan versus HIV, curious as to why included in some of the combined products.
Well, sure. Thanks for that question. It's one we get frequently. So carrageenan is, again, it was originally for HIV prevention, and around the time that the CareGuard trial was wrapping up, John Schiller and Chris Buck at NCI, National Cancer Institute, found that carrageenan is one of the most potent HPV inhibitors known. So as it turns out, by including carrageenan in our formulation, we picked up this very potent anti-HPV activity, which is which is a good thing for the product because gives it that broad spectrum MPT activity spectrum. It also turns out that carrageenan has some very nice physicochemical properties. It imparts a very nice kind of properties, viscosity and spreading properties to the gel formulations. So we use carrageenan for a couple of different reasons. One for its delivery attributes and another for its potent anti-HPV activity. We also found, again, another bonus of using carrageenan is that in our MIV-150 zinc acetate carrageenan gels or products, which is our PC1005 series, we found that zinc acetate and carrageenan have synergistic HSV2 activity. So the combination of zinc acetate and carrageenan has much better activity than zinc acetate alone or carrageenan alone. Interestingly enough, we also found the same thing with our PC6500 series. We found that Grifficin has kind of moderate HSV2 activity. Carrageenan, again, has moderate HSV2 activity, but if we mix the two together, we now get a very potent HSV2 product. So again, for reasons of physicochemical properties, HPV activity, and the synergistic HSV2 activity, we really like carrageenan and try to include it in every product we have.
Thank you, Tom. And I'm going to throw in one of my questions because it relates a little bit to this gel here. When you described the gel, a combination of Grifficin and carrageenan and the FDI, or fast-dissolved insert, you described the gel as being thick and viscous. And so I'm trying to understand exactly how thick and viscous you're saying. Is it, it sounds to me like it could be too much and it might mess up the natural lubrication. So can you just describe a little bit more about how that gel interacts with the vaginal lubrication naturally and how that thick viscous is for people?
Well, sure, Jim. So I don't have it's hard to describe just how thick and viscous it is, but let me go back to one other property of a carrageenan-based gel, which is that it's a so-called shear thinning gel. So carrageenan gel has a certain viscosity and certain spreading properties, but then in the presence of a stress, and it's well known that sexual intercourse is a stress. Under stressful conditions, whether we do it in a laboratory or during sex, those carrageenan-based gels, they kind of relax. They get less viscous, they spread more, and then when sex is over, when the stress is over, they kind of come back to their original form. So even if we have a viscous gel, which is formed now by vaginal fluid dissolving the FDI, during sex, that gel is going to thin a little bit. It's going to spread throughout the vagina, and it's going to protect from HIV and STIs. And again, once sex is over, then the gel will kind of return to its original viscosity, and it's not going to be leaky or messy.
Okay, thank you so much, Tom.
Sure. And one more thing, Jim. Remember, vaginal fluid, a typical gel dose might be 4 milliliters of gel. Typical vaginal fluid in a woman is going to be about 1 ml to 1.5 ml. So that's the amount of gel we're going to have be produced by the FDI versus 4 grams, 4 milliliters with a gel dosing. So it's a big difference.
Okay, that's very helpful. Thank you. My mind was going a number of places when I heard thick and viscous.
Well, sure. No, glad I could clear that up because it's probably a question more people than you had. Yeah.
Maybe, yeah. So going back to the chat, we have another question. What is the non-hormonal contraceptive in the oral formulation with Grafiston?
Yeah, we're not quite ready to disclose that. Sorry. We've got a couple of funding applications in, and once we receive the score and receive the funding, then that'll all be public. But for now, we prefer to keep that under wraps.
Okay, fair enough. Got you. So another question about the Truvada combination. Can you explain a little bit more about your timeline and approach for the Truvada levonorgestrone products?
Let's see. This is a multifaceted question. So the timeline really depends upon funding. So we already have, as Tom mentioned, we have some money from the SIP Foundation to work with a contract group who's, as I say, coming up with the recipe of exactly how to put together Truvada with a combined oral contraceptive. So, the 2 pills separately have their own excipients, their own formulation, and then, someone has to figure out, so we have contracted with somebody to figure out what is the recipe when you put these things together so that they're still stable, et cetera. So that work is already happening, but in terms of, the real push, what we would need is bioequivalence studies. So those are clinical trials. So even though, no, we don't need, a phase 3 clinical trial, ultimately we would need some kind of bioequivalence study to show that if these 2 products are in one pill, they work the same way as if someone took the 2 pills independently. So really it's a matter of getting the funds necessary and the buy-in that this would be something that people would be interested in and that donors would be willing to purchase to give to women, especially in places where adolescent girls and young women are really at high risk of both HIV and unintended pregnancy. So part of our work, in addition to the product development piece of trying to come up with this recipe, is also doing market research and to see what the market would be, both in the private sector and the public sector, as well as something that we have not yet undertaken but intend to is an impact analysis, like what would be the impact of such a product on rates of HIV and unintended pregnancy in specific areas where those are the biggest concerns. So it's hard to, I think if we had all of the money that we needed to do all of this, it could be 3 to 5 years to have something out there that people could use.
Thank you, Barbara. I'm going to pause again and see if anyone wants to ask a question verbally. You'll *7 to unmute your phone, identify yourself, and ask your question or make your comments.
Hi, this is Anna Forbes. I had another question, if that's okay.
Go ahead, Anna. Yes.
Yeah, I remember years ago when we first started talking about Griffiths, and it sounded as though it might be an incredibly good idea because it was something that women living with HIV could use with no risk of developing resistance to another ARV. So in a combination deal, presumably they would get contraception and hopefully some protection from strains of HIV that they don't already have without developing resistance. Am I still remembering that correctly, and is that still the case in your opinions?
Yeah, that's That's true. We could, Griffithsin has broad-spectrum antiviral activity against numerous clades of HIV, and that seems like it would be totally appropriate. And one thing I didn't mention about the Griffithsin-based products is that there wouldn't be a need for HIV testing. So someone, man or a woman could use a Griffithsin-based product if they didn't know their HIV status, number one. And once we were able to show that the Griffithsin product is both stable and safe in a kind of under a prescription regimen, go to a doctor, go to the pharmacy type regimen, then we anticipate eventually it could be on a OTC status. So I got my acronyms messed up. Over-the-counter status at a pharmacy. So it could be potentially a much easier way of getting HIV and, in some cases, HIV and contraception protection than going to a doctor or a pharmacy.
Well, I just want to say that I think that for those of us in the advocacy world, which I know is not your particular profession, Tom, this is something I think we should be making a whole lot of noise about because the possibility of having something that could be used, particularly if it could be marketed OTC, and that it's effective for contraception and helps to reduce HIV risk, whether the person is positive or not, in the sense of reducing risk of getting a new strain of HIV if you are positive. That's just amazingly good news.
Well, thank you. And, we look forward to everyone helping to spread that message.
Thank you. So I'm going to go back to the chat and combine a question that is a question I also had. So someone is asking, aside from the NIH, are there other donors that could be encouraged to invest in MPT R&D? And I want to just add a little friendly amendment to that to ask you both what your thoughts are, kind of what your prognosis is for the future. You have a lot of really interesting things in the pipeline. Many of them are very early phase or animals or early human studies, and are going to obviously require lots of investment to get them across the so-called finish line. And with what we know about the NIH's priorities, what we have seen with some of the cracks that we have opened up in the FOA, what are your thoughts on funding for the future, both NIH and, to this person's question, aside from the NIH?
Okay, so this is Tom. So I'll start off. I'm not sure we want to necessarily rule out NIH completely because NICHD, so the National Institute of Child Health and Development, has recently put out a number of FOAs that are kind of right up our alley. They wanted, one of them seemed like it was almost written for us. They wanted on-demand, non-hormonal contraception product. And they also wanted with STI prevention, so anti-infective properties. So you can imagine how our Griffithsin carrageenan non-hormonal contraceptive program fits right into that. And again, I think there have been 2 different programs from NICHD that have addressed this on-demand non-hormonal products. So we jumped on that immediately, and those are two of the applications that we're currently waiting on, and will receive scores sometime in late March, early April. So NICHD seems to be picking up really kind of the ball for us in terms of moving our products forward. USAID, and I neglected to mention them on the slide about the FDI. They supported our microbicide work for at least 12 or 15 years. Yeah. They were early supporters of our PC 1005 program, the MIV-150 zinc acetate carrageenan, and they fully supported our PC 6500 program. In fact, without their funding, we would not have a PC 6500 program. So we want to really thank them for their long-term support of both programs. And I think before my voice gives out, I'm going to turn it over to Barbara.
Thank you, Tom. Although I'm not sure that I have much else to add, I think the funding climate is very difficult, and I think there are certain funders who believe that long-acting systemic methods are the way to go, and so trying to keep up the the mixture and the interest and the support of multiple donors is definitely a challenge for all of us. In the past, we've also sought funding from DFID. Now, as Tom mentioned, we have this money from SIF to begin the oral CMP development. So we're looking at other funding sources, and we're also hopeful that USAID will be able to continue supporting us. Tom said the last 15 years, but actually it's been even longer because USAID basically and Gates were the ones who supported our CARAGARD phase 3 trial. So I think, I don't know that we have any words of wisdom except that we have these really exciting products, and hopefully we'll be able to get some traction with funders to keep them going.
Thank you. Now, I know we're at the hour. Do you have a few more minutes to get a few last questions? Otherwise, we can do follow-ups with them. Are you okay with that?
Yeah.
Okay. So we know at CROI coming up, it looks like we're going to have a presentation on FTAF or Descovy from Gilead, which has tested it as a daily prevention pill. Has there been any discussions at Pop Council about a co-formulation with TAF and the contraceptive, TAF or Descovy and the contraceptive?
Well, the short answer is yes, and that's all we can say right now.
Great.
Okay.
Thank you.
It hasn't escaped our. It's a fairly obvious thing, and we're in discussions.
Okay. Fair enough.
All right.
Very good. Going back to the gel, the viscous gel we were discussing. A person asked, if the gel firms up again after sex, does that mean it stays in the vagina longer?
No, it eventually washes out. I think we have some PK data showing after 24 hours there's no more carrageenan left in the vagina in vaginal lavages. So I think that's maybe that's the time frame we could be talking about. It's not there permanently or for long duration.
Great. And then I think our final question, and then I'll do a quick wrap up. Is the regulatory pathway for an FDI clear, and how long are the different phases?
Yeah, well, that's a multifaceted question.
Indeed.
Well, certainly we have a pretty good idea about phase 1 because we've had a pre-IND meeting with the FDA and we received some written answers. So we know what we need to do to get the Griffiths and carrageenan FDI into a phase 1 trial. After that, we still have a pretty good idea because I think it would be a fairly standard development program. I think really the key might be at some point we're going to have to pick a pathogen, so to speak, and decide whether we want to focus the initial IND on, or initial efficacy testing on HIV, HSV2, or HPV. And depending on what we decide could change the development program. So I think at this point the phase 1 strategy is set, but phase 2 and phase 3 I think is still to be decided.
Thank you. Well, I want to again thank Pop Council and Barbara and Tom for this great discussion. And I'll just want to mention again, we had a really large registration for this call, over 100 people. That does not happen very often. And I think it really just speaks to the interest in these products and the work that you're doing. And I really hope that this interest translates into adequate, appropriate support from all of our funders, NIH included. And I will again say that none of this happens with us sitting on the couch and wishing and praying. We need to make our voices heard. We need to be engaged. We need to be talking to people. We need to be demanding a full array of prevention methodologies, from long-acting to short-acting, from things delivered in the clinic to things delivered by yourself when you need them, for different people during different phases of their life. It seems all really obvious, but I think we need to bang that drum and those drums over and over and over. Take every chance you get. Look forward to having these slides sent to you shortly. In the next couple days, the slides and the recording. We'll send you a link to these resources. We hope to continue this conversation. IRMA and AVAC are interested in doing more webinars like this that are showcasing the entire breadth of the HIV prevention research pipeline, which is very, very exciting and interesting. Lots of interesting stuff in early phase studies that we would like to see appropriately resourced as things move forward beyond 2020 and 2021. And I again want to thank AVAC for all their wonderful support, and Levi Butcher in particular for all the behind the scenes magic that he makes. So I will now close out from sunny, beautiful Beach Day, Chicago to wherever you are. Have a wonderful day, wonderful evening, and we'll talk soon. Bye-bye.
Thanks, everyone. All right.